This section explains the single generative idea behind Mendel's principles of heredity. Blending was the wrong ontology. Mendel's move was to see that the smooth continuum of inherited traits is generated by discrete, conserved particles, 2 per trait, 1 from each parent, that separate at gamete formation and repair by chance at fertilization. The famous 3-to-1 ratio is the arithmetic shadow of that hidden combinatorics. Count the offspring and you read off a mechanism you cannot see. This is the whole generative primitive the book unfolds. Phenotype ratios are the observable projection of a particulate probabilistic bookkeeping. Everything downstream, chromosomes, DNA, genomes, fills in what Mendel could only posit as an abstraction. An imagined particle that worked by an unknown mechanism. This section explains why heredity has to be built from discrete units and how Mendel proved it. Blending inheritance makes a testable prediction and then fails it. If parental traits fused like pigments, variation would average away within a few generations. Yet siblings differ and lost traits resurface. That single failure forces the chapter's thesis: heredity is particulate. Traits are carried by discrete units that pass intact, recombine, and separate rather than merge. Mendel's move is the deep one. He inferred the existence and the discreteness of an unseen carrier purely by counting clear-cut alternatives across generations in pea plants, reading a hidden alphabet off phenotype ratios. The primitive to keep is this: a quantized substrate recovered from the statistics of visible outcomes decades before anyone could see the gene itself. This section explains how the extensions to Mendel's laws all arise from perturbing a single map. Mendel's laws describe transmission, how alleles segregate and assort into gametes, and every so-called exception in this chapter leaves that machinery untouched. What bends is the map from genotype to phenotype, which Mendel took to be 1G, 2 alleles, clean dominance, 1 trait. Relax. Each assumption independently and you regenerate the whole catalog. Incomplete dominance and codominance, relax the dominance relation. Multiple alleles, relax the 2 allele count. Pleiotropy makes 1 gene write many traits. Epistasis and gene interaction make traits read many genes. The primitive is the factorization. Transmission stays Mendelian, and the phenotype function is what varies. So any new ratio you meet is just another perturbed mapping over intact 3-to-1 arithmetic. This section explains why the messy inheritance ratios of Chapter 2 never actually break Mendel's laws. Mendel's laws govern transmission, how alleles segregate and assort into gametes. They say nothing about how a genotype becomes a phenotype. That second map is where every deviation lives. Chapter 2. Pre's the 2 layers apart. Incomplete dominance, codominance, multiple alleles, recessive lethals, gene-by-gene interaction, and environment. Each one complicates the step from genotype to phenotype, while the transmission machinery underneath stays exactly Mendelian. The messy ratios that seem to break the laws never touch them. They decorate the readout. Here is the primitive to keep. When a clean 3-to-1 ratio fails, do not ask whether segregation changed. Ask how the genotype cashes out into the phenotype. The fault is always in the mapping, never in the inheritance. This part explains how the gene stopped being a bookkeeping token and became a physical object you can see. Mendel's laws were bookkeeping over invisible tokens. Segregation, independent assortment, fixed ratios, all of it accounting with no object attached. This chapter supplies the object. Chromosomes move during cell division exactly as Mendel's genes behave, so genes must ride on chromosomes. The whole argument is one move, an isomorphism between an abstract combinatorial law and a visible mechanical process. You identify the carrier of a hidden quantity by matching its behavior to something you can watch under a microscope. That converts the gene from a counting fiction into a physical thing that can be seen and cut. Trisomy 21 is the tell. One extra copy of a normal chromosome deranges the phenotype. Inheritance depends on how much material, not only which kind. This section explains how an abstract law of inheritance gets welded to a physical object you can see under a microscope. Mendel gave a law without a mechanism, invisible factors that segregate and assort. This chapter supplies the missing referent. The factor is a stretch of chromosome, and meiosis is segregation made visible. The argument is pure isomorphism. Two systems are described independently: the bookkeeping of alleles across crosses and the choreography of threads under the microscope. They obey the same combinatorial law, so they are the same thing. Homologs part at meiosis 1, and that parting is the segregation law. Different chromosome pairs orient independently, and that independence is assortment. Once the behaviors coincide exactly, identity is forced. The abstract particle collapses into a body you can stain, count, and lose, as in trisomy 21. This part explains why sex became the trait that turned abstract heredity into something visible. Sex is the one Mendelian character whose hereditary factor you can actually see. Every other character hides in an abstract element. Sex hides in an entire chromosome, the unmatched XY pair, one large and one small conspicuous under the microscope, where the other 22 autosomes come in matching pairs. That visibility is the whole move. Sutton watched grasshopper sperm split the X from the Y in exactly the 1-to-1 ratio that Mendel's factors demanded. The invisible law suddenly had a physical object obeying it. This chapter is the book's bridge, the trait that anchors abstract heredity to a thing you can stain and count. Everything downstream, sex linkage, the physical mapping of genes, rides on this one coincidence of a hereditary factor large enough to see. This section explains why sex is the visible anchor that gives a Mendelian factor a physical address. Start from a rule of inheritance. A trait whose two forms are mutually exclusive and surface in a 1-to-1 ratio is the phenotypic fingerprint of a single segregating pair. Sex is exactly that. The chapter's move is to catch that abstract pair in the act of being a visible object. Males carry one unmatched pair, an X and a Y, where females carry two matching X chromosomes. Once an either-or phenotype at 1-to-1 lines up with a chromosome, you can see down a microscope. Mendel's bookkeeping factor acquires a physical address. That is what sex chromosomes are for here. They are the welding point where the gene stops being an abstraction and becomes a location you can stain, count, and follow through meiosis. This section explains how genetic linkage turns a broken law into a measuring instrument. Genes riding the same chromosome betray Mendel. They travel together instead of assorting independently. Crossing over occasionally cuts and swaps the strand, and the chance the cut lands between two genes grows with the gap between them, so the fraction of recombinant offspring becomes a ruler. That is the chapter's whole move, Sturtevant's leap. A counting statistic read off progeny is silently a spatial coordinate along the chromosome. Define 1% recombination as one map unit, and linkage becomes cartography. Everything downstream, 3-point crosses, chi-square tests, genetic maps, is bookkeeping on this one translation of event frequency into distance. The failure of a law is repurposed as a measuring instrument. This section explains why linked genes give genetics its rulers. Independent assortment fails whenever two genes ride the same chromosome, and its failure is the ruler. Linked genes travel together. Crossing over during meiosis occasionally severs them, and the severing rate climbs monotonically with the physical gap between them. So a statistical rate becomes a spatial coordinate. Count the recombinant progeny, and you have measured a distance spatial coordinate. Count the recombinant progeny, and you have measured a distance. Chapter 5 is where genetics earns its maps, the pedigree showing color blindness clinging to hemophilia. A yet drifting free of hemophilia, B is already reading position off frequency. The primitive to carry forward, when an observable behavior varies monotonically with a hidden quantity. The behavior is a measurement of that quantity. This section explains how a single geometric fact organizes the whole chapter on DNA structure, replication, and recombination. One fact carries all of it. Each base pairs with exactly one partner, A with T, G with C. So the two strands collapse into a single fact, each the negative of the other. From that single complementarity, everything follows without added machinery. Sequence stores information because the pairing rule leaves base order free. Replication copies faithfully because each strand dictates its partner, acting as a template rather than a blueprint. Recombination exchanges strands because homologous sequences can repair across molecules. The chapter's framing is flat. DNA's genetic functions flow directly from its molecular structure. The three nouns in the title name one structural consequence viewed from three angles. This section explains why one geometric fact about DNA gives you the whole chapter. Structure is function here, and the entire chapter collapses to a single move. Complementary base pairing, A with T and G with C, makes each strand the exact template of the other. From that one geometric fact, three capacities fall out with nothing further added. Sequence stores information. An unwound strand dictates its own copy. An unwound strand dictates its own copy, and a broken region rebuilds from its partner. Replication, storage, and recombination are three readings of the same complementarity, not three properties bolted onto DNA. The molecule is chemically inert on purpose. Its power lives in shape rather than reactivity, which is exactly why it can archive information for 38,000 years. Learn the pairing rule and you regenerate the rest. This section explains why a single number, the rate at which DNA miscopies itself, drives the whole story of mutation. One quantity generates the entire drama. Mutation, heritable change in base sequence, is the sole source of the variation that selection acts on. Yet the same fidelity that would abolish mutation would also abolish evolution. The chapter's single thesis is that this rate is tuned. It is held at a chosen value rather than driven to its minimum. Near-perfect repair pushes it down, and residual inefficiency holds it just above zero. So the marvel is negative. Lewis Thomas called it the capacity to blunder slightly. The locus that stores information also stores the potential for its own revision at the same address. Everything downstream, the allele, the phenotype, and genetic analysis itself is a consequence of one small defended error rate. This section explains why mutation is a residual quantity rather than a fixed property of DNA. Mutation is a residual. It is the small remainder left when two opposing processes run against each other. On one side, damage constantly generates base changes. Indices developed in Chapter 7, from radiation, from replication error. On the other side, repair constantly reverses them. The observed mutation rate is only the small leak between the two. The observed mutation rate is only the small leak between the two, and it exists because repair is highly efficient. This is developed in Chapter 7. That gap is the whole thesis. Too much leak dissolves a species' integrity. Zero leak forecloses adaptation. So life balances on the narrow rate, where correction almost, but not quite, wins. Lewis Thomas names it exactly. The capacity to blunder slightly is the real marvel of DNA. This is the same primitive as any imperfect error-correcting channel, where the uncorrected residual is precisely where novelty lives. This section explains how mutations let you study a gene you can never see directly. You never see a gene. You see only its broken variants, and the way two lesions interact tells you the structure behind them. That is the whole move. Perturb an invisible unit and read the pattern of perturbations. 2 operations. The first operation puts 2 recessive mutations in trans. If wild type is restored, they sit in different functional units. If not, they are the same one. This gives you the gene as the unit of function, and the test is called complementation. The second operation asks whether a crossover between 2 same gene lesions can regenerate wild type. It can. So the unit is internally divisible, the gene as a set of independently mutable, recombinable base pairs. The Mendelian bead dissolves into addressable interior. Read entirely off damage. This section explains how you learn the shape of a gene you can never see directly. A gene is invisible. You learn its shape only by breaking it in many ways and reading structure off the pattern of breaks. That is the whole chapter. Perturbation is the instrument. 2 independent readouts fall out of the same trick. Complementation asks whether 2 mutations, placed on opposite homologs, can cover for each other, defining the gene as a unit of function. Recombination asks how often 2 mutations separate, defining it as a unit of divisible structure. Together, they dissolve the bead on a string. The gene turns out to be a stretch of independently mutable, independently recombinable base pairs, rather than an indivisible atom. You never see the gene, only the silhouette its lesions cast. This section explains gene expression as the flow of information from DNA to RNA to protein. A gene is a channel whose sequence gets rewritten twice, and each rewrite is the same trick. A template pairs complementary bases against itself. Transcription transliterates DNA into RNA. Same 4-letter alphabet, base, swapped, still invertible by the pairing rule. Translation then crosses alphabets, reading messenger RNA in fixed triplets against the genetic code, mapping 64 codons onto 20 amino acids. That second map is the break. Many codons to one amino acid has no inverse. So, as Chapter 9 puts it, once information has passed into protein, it cannot get out again. The whole chapter is this one asymmetry. Complementary pairing moves information losslessly right up to the codon dictionary, where a many-to-one lookup makes the flow irreversible. One gene, one polypeptide, because the channel is discrete. This section explains how a gene becomes a protein, and the one primitive that makes it possible. Genetic expression is a translation between 2 alphabets that share no symbols, 4 nucleotides on one side, 20 amino acids on the other. What makes such a translation possible is 2 things working together. First, Order is preserved, meaning a gene is collinear with its protein. Second, a fixed lookup table. The genetic code maps each nucleotide triplet to exactly one amino acid in the same way that Morse maps groupings of dots and dashes to letters. Crick's central dogma fixes the direction of this flow: DNA to RNA to protein, never backward. As Crick put it, once information has passed into protein, it cannot get out again. Everything downstream serves this one rule. The copying, done by transcription, the translation carried out on the ribosome, the transfer RNA adapters, all of it is machinery serving a single primitive. Sequence determines sequence, one way, through a code,